Hello, everybody. My name is Paolo Caliceti, and I'm full professor at the University of Padova in the Department of Pharmaceutical and Pharmacological Sciences, and where I teach pharmaceutical technology and drug delivery. And I work since several years in PEGylation of proteins. And in the next few minutes, and with the next slides, I will provide a few information about the biopharmaceutical and immunological properties of PEGylated proteins. Please, I remember if you wanted to ask questions, to use the click on the bottom below, as was mentioned by Professor Gonçalves before. Well, what is PEGylation? We heard by Professor Gonçalves the general introduction of PEGylation. And PEGylation is a mature and unique technique to produce new protein conjugates, new protein derivatives. Since about 50 years ago, when it was proposed by Abukowski and Davis, who worked at Rutgers University in the USA, PEGylation of protein became a mature technology that provided for a great solution for the delivery of proteins. The new class of drugs which were developing at the end of the past centuries, which had a lot of problems in formulation because they are big molecules, very fragile, they undergo degradation, denaturation, and whatever. So PEGylation provided a good solution for their formulation and delivery. PEGylation is a technology which produces new drug entities by de facto, because it is the chemical conjugation of a polymer, polyethylene glycol, to proteins. And since there is a covalent conjugation, the result is a new molecules where the polymer and the proteins are covalently bound to each other. So there is the possibility to obtain a number of different bioconjugates which are not similar each other, because their structure and their performance in vivo and the biological activity and biopharmaceutical properties depend on the polymer, depend on the proteins, depend on the number of polymer chains, shape, and the length of the polymer, the conjugation site, and the chemistry of conjugation. So there are not properties of one PEGylated protein that can be extrapolated to another. But of course, we can have a general indication about the effect of polymers of PEGylation on the final bioconjugate. So what we have in terms of characteristics of PEGylated proteins and how their properties are affected by the conjugation of the polymers. By the protein stand view, there are several modifications of the performance of the proteins after polymer conjugation. First of all, is the permanence in circulation, and that was the first main problem, which was in some way overcome by PEGylation. Actually, proteins are rapidly cleared when their size is below 60,000 Dalton, which is the threshold for glomerular filtration. And in the case of PEGylated proteins, the proteins has a prolonged permanence in circulation. And this is due to a number of different effects, but the main effect is related to the increased size of the bioconjugate compared to the native protein. This is due to the fact that the PEG is a very hydrophilic polymer. Each monomer coordinates 2 or 3 water molecules. And this has as a result the fact that the polymer is hydrated and the hydrodynamic size is much larger than the physical size and the duration radius. Accordingly, the PEGylated protein has a larger size and larger hydrodynamic radius, and this prevents the ultrafiltration. This is, of course, not the only mechanism according which the circulation is prolonged. And also the ultrafiltration by kidney is reduced because we usually think that ultrafiltration is just a simple filtration, but actually there are many other mechanisms which include the structure of the protein, the uptake, reuptake, and interaction of the protein with the glomerular tissue. And in this case, it is also maskable. So in terms of biodistribution, PEG and elimination, PEG has a strong effect on the protein profile. The kidney is the main way the main way for elimination of the protein. And that's depend on the mass of the polymers which is conjugated to the protein. And the mass of the polymer depends on the size of the polymer chain and the number of polymer attached to the protein structure. When the polymer is very small, and we can say that the PEG with a molecular weight lower than 20 kilodalton, the elimination is quite rapid. And it decreases as the PEG increases in size and in ramification. So as higher is the mass of the polymer on the protein surface, slower is the kidney filtration. High molecular weight PEG is also dictate also the accumulation of the conjugate, even if in low amount in liver and in the lymphatic system. And also PEG has an effect in what is called the cellular vacuolization, and where there is the formation of vacuoles into the cells, which is, however, is not a permanent effect, and that they disappear throughout the time. PEG has also a great relevance in protein stability, either for what concerns the physical stability of the proteins and the stability towards proteases. In terms of physical stability, PEG, which is a very hydrophilic material, prevent the association, aggregation, and also the absorption on the surfaces of primary packaging. And in terms of protein stability towards proteases, the polymers cloud around the protein, prevent the approaching of enzymes and the recognition of the sequences which are prone to degradation. So there is a protection from degradation. Finally, the PEG on the surface also reduces the immunogenicity of proteins, especially non-homologous proteins and xenogenic proteins, which are often used in therapy. And so the immunogenicity is decreased because the protein is not recognized by the immune system. And there is also a reduction in antigenicity because there is a lower interaction of preformed antibodies which recognize the protein because the protein is protected into the core of this conjugate. As a result, we have an increased stability and prolonged permanence in the body. So the benefit of the PEGylation is well represented by the example of interferon. Interferon is a drug with a high effect and therapeutic activity against hepatitis C virus. The traditional therapy was a frequent administration of this cytokine, which was at least 3 administration per week. Because and that was due to the fact that this protein, which has more or less a molecular weight around 20 kilodalton, is rapidly cleared from the body. So it was necessary to boost the administration. Unfortunately, the frequent administration of this interferon had a very high side effects, adverse effects, and there is in terms of pain, in terms of headache, high temperature, and that was very high debilitating and prevented the therapy in some way. So the result that was obtained with PEGylation, it was to obtain new interferons which have a much lower side effect and could allow for a single administration per week. We can see here the pharmacokinetics of the native interferon in green, and in colored we can see two different PEGylated interferons. With the pink, we can see a interferon PEGylated with 40 kilodalton branched PEG, and we can see the permanence in the bloodstream of the conjugate, and in light pink we can see the activity of this conjugate, which slowly decreases throughout the time. In light yellow, we can see the pharmacokinetics of interferon modified with linear 12 kilodalton PEG. And in orange, we can see the activity of this conjugate, which decreases throughout the time. So the PEG structure affects the properties of PEGylated proteins, and we have different PEGs that can be used for the protein conjugation, and they are different by molecular weight, shape, and also we can have different conjugation chemistries. And also we can obtain derivatives with a different number of PEG chains attached to the proteins. As a result, we obtain derivatives with a variable mass of PEG conjugated to the protein, and the result is extremely different one by one, so each other. So we can have a single linear PEG with different molecular weight, which can be attached to a single site of the protein. We can have also a branched PEG, and that was used, for example, for interferon, and in such a case, it was a 40 kilodalton PEG, which is attached to one site of the protein. We can also have a single branched PEG with 3 arms. It is an hyperbranched PEG. We can have several PEG chains attached to the protein, and also in a very few cases, we can have also cross-linked dimeric PEGylated proteins, which are obtained by using PEG, which is functionalized to both of the end of the polymer chains. So the conjugation of this PEG have different effects in terms of permanence in the bloodstreams, and in terms of stability of the protein, in the immunogenic profile of the proteins, and also in terms of activity. For example, as reported here, the attachment of 40 kilodalton PEG branched to interferon, produce a derivative which maintains only 7% of the activity of the native protein. This is a very high decrease of activity, which is compensated anyway by the excellent performance in terms of permanence in the bloodstream and in terms also of reduced side effects. We have several clinical advantages of PEGylation. As I mentioned before, PEGylation can increase the half-life of the protein by up to 20-fold in humans. And we can see here the case of PEGylation alpha. And we have in these derivatives, we have 4 PEGs with a molecular weight of 2 kilodalton, which are conjugated to the proteins, which is used in the Fabry disease, and there are 2 molecules of proteins attached to these polymers. And the result is an increase of the permanence of the proteins from 3 hours to several hours, between 53 to 121 hours. There is the protection from proteolysis, which has been observed in mice, and there is an increase in thermal stability and slow down diffusion. And also this is used to have a prolonged permanence in the bloodstream when it is administered by intramuscular or subcutaneous administration. And then PEGylation decreases the immunoreactivity of the protein, and this allows for the administration also of non-human proteins, such as the case of asparaginase, yes, and adenosine deaminase, which are not human proteins. And their use was limited by their high immunogenicity, and the use of polyethylene glycol reduced the immunogenicity of the protein, and it allowed multiple administration. So the potential drawbacks of PEGylation, as I mentioned before, is a decrease of bioactivity that depends on the protein and the protein activity, and also on the mechanism of activity and the PEGylation process, the mass of the protein, the polymer attached to the protein. The dramatic effect of conjugation of 40 kilodalton PEG to interferons, which reduces the activity of 93%, is not so high in many cases, and there are techniques to prevent this high decrease of activity. One reason of the reduction of activity is the conjugation of PEG to amino acids, which are involved in the biological activity of the proteins. So one strategy to reduce this decrease of bioactivity is to conjugate PEG to amino acids which are far from the active site of the protein. Another drawback is the PEG, anti-PEG antibodies, which in a few cases represent a limitation for the long permanence of the proteins. Anti-PEG antibodies can be generated by administration of PEGylated proteins, but in many cases they are already present in the organism because they are produced by the use of cosmetics or other products. And then there is the immunoreaction to alter the protein conformation, because PEGylation is a chemical conjugation to protein, and this chemical conjugation to proteins can result in alteration of the protein structure, and the protein could move to be more immunogenic.